In this experiment, one of the most important biological processes, photosynthesis, was observed. This is shown in the equation here. Photosynthesis, occurring in the chloroplasts of plants, allows light energy to be converted into chemical energy, stored in sugar and other molecules. The rate of this process can be measured by the production of oxygen, a byproduct of photosynthesis. In this experiment, leaf discs will be placed in a carbon-containing solution, causing them to photosynthesize and produce oxygen, which will then cause them to float. The rate of photosynthesis will be compared between spinach and lettuce leaves, with the independent variable being the type of leaf and the dependent variable being the amount of leaves that float. It is hypothesized that if both spinach leaf discs and lettuce leaf discs are placed in a bicarbonate solution, then the spinach leaf discs will float more.
Now for the materials. For this lab, you will need 2 plastic cups, 200 milliliters of bicarbonate, 10 discs of spinach leaves, 10 discs of lettuce leaves, 2 12 milliliter syringe and syringe caps, tweezers, 3 hole punch, lamp with Sylvania 800 lumen LED bulbs at 5000 K temp, paper towels, timer, permanent marker, and ruler. Now for the procedure. Step 1. Label 2 cups lettuce and spinach. Repeat this with syringes as well. Step 2. Activate the spinach and lettuce by spraying it with water and placing light above them. Step 3. Pour 200 milliliters of sodium bicarbonate solution and pour roughly 3 centimeters into both cups. Step 4. Using a single hole punch, cut out 5 leaf discs from both spinach and lettuce. Step 5. Place the leaf discs into appropriate syringes and add a small amount of sodium bicarbonate solution to each. Step 6. Place the cap on the syringe and then draw back to create a vacuum, and swirl to keep leaf discs suspended in the solution. Step 7. Release the vacuum, causing the leaf discs to sink. Repeat step 5 up to 3 times until all the discs sink. Repeat steps 4 through 6 for the lettuce.
Step 8. Add fresh sodium bicarbonate solution to a clean 200 milliliter beaker to a depth of approximately 3 centimeters, and add the contents of the syringe labeled spinach to it. Repeat the process with lettuce.
Step 9. Transfer the leaf discs and the sodium under a light source that is approximately 8 inches from the light. Step 10. Start the timer. After each minute, record the number of floating discs. Swirl the discs to release any that are stuck against the side of the cups. Continue for 10 minutes.
Our data is shown here. As you can see, the lettuce seemed to show no change in the sodium bicarbonate solution throughout the 15 minutes, meaning that 0 of the lettuce leaf floated to the surface during the time period. The spinach, however, responded quite differently. The spinach started to rise at 0 discs coming to the surface, but then as the time elapsed, they began to rise. By the 5-minute mark, 2 out of the 5 discs had risen. At the 10-minute mark, 3 discs had risen. Finally, at the 15-minute mark, 4 discs had floated up. It was also noted during the experiment that the lettuce leaves were a much lighter shade of green than the spinach leaves.
In our experiment, it was hypothesized that if both spinach and lettuce leaf discs are placed in a bicarbonate solution, then the spinach leaf discs will float more. This hypothesis was validated in the lab, and by looking at the data, the gradual increase of the floating spinach discs as time elapses can be seen. This, however, did not apply to the lettuce discs. Though the data supported the hypothesis, there were potential errors that could have skewed the results, causing leaf discs to sink. For example, as the lettuce leaf discs were a much lighter shade of green, there may have been less chloroplasts in the leaves, causing less photosynthesis to occur. To prevent this in the future, fresher lettuce should be obtained. In addition to this, only 5 leaf discs were used in the experiment. If the lab were to be replicated, a larger amount of leaf discs would produce more accurate results. It would also be beneficial to have extra leaf discs just in case some do not initially float, ensuring that there is a larger sample size.